Hi, Assalamualaikum. Our title of the article is Investigation on the Biofilm Eradication Potential of Selected Medicinal Plants Against Methicillin-Resistant Staphylococcus aureus by Lakshmana Prabhu, Uma Maheswari, and Grace Felsia. Now, if you want to read this article, you can scan the QR code. Now, let's go over to the general details. What does it tell us? It mainly revolves around biofilm formation and its antibiotic resistance, which can cause life-threatening infections and also cause economic damage, which we need new approaches on. Now, the author stated that medicinal plants can be used as alternatives, such as Azadirachta indica, Moringa oleiferae, Murraya koenigi, and Pseudium guajava. They also found out that the specimens portrayed 60% to 83.9% inhibition, and it does not harm human cells. How do they know? By hemolytic activity test. Now, they also stated that the Azadirachta indica and Pseudium guajava are the two most prominent specimens which needed to be further studied upon. Now, moving on to my colleague.
This article is all about the potential of selected medicinal plants as new alternative against methicillin-resistant Staphylococcus aureus. Biofilm are multi-species bacterial communities with complex structures, and since they create antibiotic resistance that will lead to chronic infection, plants such as Azadirachta indica and Pseudium guajava are suggested as the treatment of MRSA biofilm infection. Hence, the purposes of this study are to investigate new compounds for defeating the microbial drug resistance, especially in biofilm structures, and to evaluate their antibiofilm potential. Also, the result of this investigation may contribute to the successful treatment of MRSA biofilm infections.
Next, I'll be presenting method used in this research. This research is done by the following method. Firstly, collection, processing, and extraction of plants. Leaves from the following plants were collected and labeled. This step is to extract 50 grams of coarse powder of the leaf for further analysis. Next, preliminary phytochemical analysis is done to analyze the presence of secondary phytoconstituent as per the early standard phytochemical test procedure. Bacterial isolate is done to identify Staphylococcus aureus with coagulase test and mannitol fermentation test from total of 27 isolates. MRSA is detected by using sulfur-16 disc diffusion method. Suspension of isolate was prepared on newly hinted agar plate at the concentration of 0.5 methylene standard. The agar plate was arabically incubated at 35 degrees Celsius for 18 hours to allow bacterial growth. MRSA is detected where the zone of inhibition diameter is equal or less than 21. In this step, MRSA was grown on mannitol-sorbitol agar medium, and antibiotic susceptibility of MRSA isolated strain was tested against antibiotics such as vancomycin and ciprofloxacin. Next, MIC of the extract was determined by the microbroth dilation method with different concentration of plant extract, which is from 2 mg per ml to 125 mg per ml. The samples were analyzed in UV spectrophotometer at 595 nanometer to determine the MIC. Small glass pieces of dimension 5 mm x 5 mm were used as abiotic surface for the attachment and incubated at 37 degrees Celsius to optimize the formation of biofilm by MRSA. The biofilm formation was observed through light microscope at different time interval. Next, anti-biofilm effect was assessed through light microscopy, and quantitative determination of bioformation was determined by crystal violet staining method. Percentage of biofilm inhibition was found by employing the following formula. 5 ml of human blood was used in this procedure. 500 ml of erythrocytes were mixed with 500 ml of 4 mg per ml concentration of plant extracts and incubated at 37 degrees Celsius for 1 hour. The supernatant, which have free hemoglobin, was collected, and the adsorbent was measured at 540 nanometer. The extracts were analyzed for its hemolytic activity by comparing them with positive control and negative controls. All tests mentioned were conducted in triplicate and reported as mean standard deviation.
As for result and discussion from medicinal plants extraction, it's shown that the extract contains phytoconstituents such as alkaloids, phenols, saponins, and others. Sulfur-16 disc diffusion method is used in detection of methicillin-resistant Staphylococcus aureus as it is more sensitive and specific. Out of 27 isolates, only one identified as methicillin-resistant Staphylococcus aureus. Susceptibility testing is performed to assess whether the antibiotics can inhibit the growth of the isolate. Vancomycin and ciprofloxacin were chosen as it is the usually used in treatment for Staphylococcus aureus infection. Vancomycin showed inhibition at 4 microgram per dose, whereas ciprofloxacin showed inhibition at 5 microgram per dose. The preliminary antimicrobial study showed petroleum ether extract of Azadirachta indica and ethanolic extract of Pseudium guajava showed a MIC value of 125 microgram per mil and MBC value of 500 microgram per mil. The ability of the bacteria to form biofilms was influenced by the increased time duration of the incubation period. At 24 hours of incubation, the planktonic cells get attached to the abiotic surface. At 36 hours, the planktonic cells start to aggregate, and at 48 hours of incubation, the complete biofilm was formed. The anti-biofilm effect of the extracts was evaluated both qualitatively and quantitatively by light microscopy and crystal violet staining method. Terpenoid and flavonoid are present majorly in both extracts, which possess good antimicrobial properties. Inhibition of biofilm formation by petroleum ether extract of Azadirachta indica 83.8% and ethanolic extract of Pseudium guajava 80% were observed, which is comparatively higher than that of the other extract. The other extract showed an inhibition percentage of around 30 to 60%. The assessment for hemolytic activity is by UV spectrophotometer. This extract were non-toxic and did not possess any hemolytic activity to the human erythrocyte. This minimal hemolytic activity could be due to the presence of saponins and phenols in the extracts.
Thank you for my friend. Now let's go over to the conclusion. Last part. They said that the biofilm by pathogenic microbes are a threat, and not many studies on biofilm formation, but there are a lot of studies on antimicrobial resistance. So further students need to focus on the biofilm formation topic. Next up is the hemolytic activity of specimens needed to be studied further because of uh, the damage that it can cause if it does not be tested out correctly. And last but not least, of course, further studies must be conducted for the 4 specimens or in biofilm formation field. All right. Thank you. That's all from us. Assalamualaikum.